Hello everybody. Now welcome again for the second topic, and that is on visual transduction in the eye. So we will have a closer look on the molecular processes that allow the transmission from light into a neuronal signal. So let's first start and have some insight how the eye is constructed. You can see here that light passes through the pupil and enters via or passes through the vitreous humor and reaches the final end of the eye, which is shown here. The final end, that is the most important part of the eye, and it's called the retina, as you can see here. The retina is a special part of the brain where we have those cells present that are able to interact with light, and their response to light will be a neuronal signal. You can see here that out of the retina, all of the signals are gathered together in a nerve, and that's called the optic nerve. There are some additional points here. There is a blind spot. It is inosphar blind because here we do not have those cells present that enable the transduction from light to a neuronal signal. And here, particularly here, is one area where we have the point of central focus, and it's also called the fovea or macula. So when ophthalmologists will have a closer look into the eye, they try to see the background of the eye and particularly the blood vessels. Here you can see two different resolutions. This is simply when through an open pupil the retina will be highlighted, particularly the background of the retina. Then you can see the blood vessels which are present here. The contrast is much better because the blood vessels are filled with a certain compound that makes it much better visible in this image here. So whenever you get an image of the background of your eye, the medical doctor can already come to some conclusions, particularly when the blood vessel doesn't seem does not look very good. That can happen, for instance, when people suffer from diseases such as diabetes. Diabetes or Zuckerkrank, one of the first symptoms that can be seen here by a visualization of the background of the eye, that the blood vessels do not look as they should actually look like. And sometimes this is the first indication that the ophthalmologist tells the patient that he or she should have a look on the blood sugar levels, because when these blood vessels look somehow changed due to some inflammatory processes going on, that could be an indication, for instance, for diabetes. So it's really useful to have these, let me say, these examinations on a regular basis, particularly when you're young, every 3 to 5 years, but sometimes this can be revealed during this examination. So let's have a closer look on the retina. The retina is at the very end of the eye. And you can see here in a further cartoon that there are several layers of cells, exactly 3 layers of cell which compose the retina. Interestingly, we will see in a minute the cells that show the response to light are farthest away but the distance here does not matter because there are only 2 layers of cells above, and the light can easily transmit through and reach those cells which are sensitive to light. So let's have a closer look on the various layers. You can see here how actually these cells are turned. Those cells which are farthest away and which are at the very end of the retina, where here also the blood vessels are situated, because these cells, they need a good supply by oxygen and by nutrients. The first layer here are actually the photoreceptor cells, and you see here 2 different forms already. These are the rods and these are the cones. We will come to this later. These are specialized nerve cells, and as we know from a nerve cell, we will also see it later, it's connected to the next layer of nerve cell via synapses, and these are the so-called bipolar cells, because these cells actually have 2 poles and they connect to the third layer, which is composed of ganglion cells. As typically for nerve cells, all signals, they leave the ganglion cells via axons, and all the axons, they get together to the optic nerve, and from the optic nerve they get into further deeper structures of the brain we will see at the end of my lecture. So here is an histological examination where people or histologists have tried to use different dyes to make the certain cells visible. You cannot see all the cells. These are the photoreceptor cells. This is the very end of the retina. These are the photoreceptor cells. You can see here, particularly, these are the nuclei of the bipolar cells, and what are not very well visible here are the ganglion cells. But what you can see here, this is and you can very nicely depict it from the image. These are the axons that are following a direction leading them to the optic nerve, as we've just seen before. So these are the main 3 layers: the photoreceptor cells, the bipolar cells, and the ganglion cells. As we already have heard, we have these more stretched forms, the rods, and we have these special forms which looks like cones. So these are the cones. And in between there are 2 additional sort of cells. So in between, somehow modulating the signal that is generated in the photoreceptor cells, are the horizontal cells, because they horizontally link several of those photoreceptor cells. And we have at the level of the ganglion cells also another cell type which links several ganglion cells, and these are the amacrine cells. But we will particularly focus on these 3 cells, and particularly we will be interested in what is going on in these photoreceptor cells. So you can see it here once again. It's a slightly different picture, starting here with the photoreceptor cells where a signal is generated. We have the bipolar cells, we have the ganglion cells, and in between we have the amacrine and the horizontal cells. So these are the 2 different forms, the 2 different sorts of photoreceptor cells. This is an electron microscope image, so with a much better resolution, but you can also see in this cartoon the specialized form that leads to the terming of these 2 different photoreceptor cells. These are the rods and these are the cones. And you may have already heard, maybe from your former education, that the rods are those cells that provide sensitivity for black and white light or black and white seeing, and the cones are those cells that provide sensitivity for visualizing colors. Rods, in German Stäbchen, and cones, which are called Zäpfchen. when you have a closer look, they have, of course, all the characteristics of a typical nerve cell. They have all cells. They have a nuclei. They have an axon via a synapse linked to the next cells, and in both cases these are the bipolar cells. But what is so specialized here are these special forms, which leads to the name of a cone and a rod. And these are, when you have a look at the structures, these are discs, and they are also called intercalated discs. You can see it here, the disc, they actually have all the same size, the old diameter. Here the diameter gets smaller and smaller, which ends up with this special cone form. So let's have a closer look. This is the so-called outer segment, which is different between the rods and the cones. In this segment we have a specialized membrane system, and you can see we have here an outer membrane, and within the outer membrane we have here intercalated discs. So we have here discs which provide a very large area of a plasma membrane, which is somehow here inside those cells. The same is actually also present, but here, as I mentioned before, the diameter gets smaller and smaller. But why is such a large membrane area here necessary? Because this helps to get a tremendously sensitivity to light, because particularly in the intercalated discs, not in the plasma membrane in the outside, but in the intercalated discs, you have a very high density of the so-called visual pigment molecules. These are actually the receptors that allow to interact with light. And the question is, how is such a receptor composed, and particularly what is happening within such a cell when light shines on such a photoreceptor cell? So what is happening when light shines on those cells? And you can imagine somehow it must be a very fast signal, because whenever you close your eyes and open it, you immediately see. So within milliseconds you can somehow estimate, you get a response to light. So there must be, of course, some ion channels involved. That's the reason why the signal transmission is so fast. But let's see what happens when light shines onto, and the same happens independent whether it's a rod or a cone, but the same happens with both. First of all, when we think of a specialized nerve cell, we would expect the resting membrane potential in the dark in the range of about, let me say, minus 80 millivolts. But interesting, you see, in the dark, the resting membrane potential is around minus 30 millivolts. So these cells are somehow in a depolarized state. As these cells are somehow in a depolarized state, we can somehow guess that channels are already open that lead to a sort of depolarization. But let's first have a look what comes, or what is going on with the membrane potential when light, as it's shown here, a bright light shines onto such a cell. And you can see here what happens is, as soon as the light reaches the cell, we do see a hyperpolarization. The cell gets more negative and end up with a potential in the range of about minus 80 millivolts. So that's the classically resting membrane potential, usually of a nerve cell, of a resting nerve cell. But here it seems to be different. In the activated state under light, these specialized photoreceptor nerve cells show a hyperpolarization. And this actually correlates directly with the intensity of a light. You can see here when a dim light in contrast to a bright light shines on, the signal, the hyperpolarizing signal we get is actually less. So this is directly related to the light that shines on these specialized photoreceptor cells. So what's going on? Why is there such a dramatic change in the membrane potential? So let's first have a next look. which sort of channels might be involved here based on the fact that the cells are in the dark state depolarized and then gets hyperpolarized. So keeping in mind on the different ionic concentrations that we have a high sodium concentrations in the outside and a high potassium concentration in the inside, we have heard that during the action potential, typically cells that allow an influx of sodium, they will lead to a depolarization of a cell. So one can guess, although it's not an action potential here, but one can guess here that maybe in the dark there are sodium channels open. You can see here in the dark we have a steady state sodium influx. When dim light, or let's have a look on the bright light. On the bright light, when bright light shines, we have heard the cells hyperpolarize. So somehow this sodium influx has to be stopped. And you can see here, the brighter the light is, the more of these sodium channels get closed. So the change from a depolarized state to a hyperpolarized state, when light shines on a photoreceptor cell, actually comes from first, in the dark state, an opening of sodium channels, which somehow, due to a mechanisms we still have to understand, close when light shines on these photoreceptor cells, which finally leads to an hyperpolarization. So let's further continue what's going on, what's happening with these specialized photoreceptor cells that these channels get from an open to a closed state. So and first of all, we are asking what at the very beginning of this cascade, what is the receptor for light? And this was one of the first G protein coupled receptors that have been identified and particularly crystallized. So this is the specialized receptor, and it's one of the first that have been crystallized, and it's a G protein, it's a classical G protein coupled receptor. It has, as all of those G protein coupled receptors, it has 7 transmembrane helices, with the start of the amino terminus in the extracellular side and the C terminus ends at the cytosolic side. But classically, a protein, we have heard that there are really exceptions from the rule that a GFP, a green fluorescence protein that can interact with light, but classically proteins, they do not have a sensitivity to light. So the nature must have done something to provide these rhodopsines or these proteins with a sensitivity, and particularly, we will see it later, a chromophore is embedded in the structure of these G protein coupled receptors. And you can see here the main point is for these receptors to get activated in response to a ligand, where we have to say the ligand here is light, is that the chromophore, as you can see it here, we will see it in a second in more detail, when it gets into contact with light, so it absorbs light, we will have a change in the conformation. And the conformational change of a chromophore is transduced to a conformational change of a whole protein. And this initiates the receptor cascade that finally in the next step involves G proteins. But let's have first a closer look on the sort of chromophores involved. So the chromophore, and the best you look at the panel below here, the chromophore involved here is called 11-cis-retinal. You can see it here, it's quite a long molecule because it somehow allows or has sensitivity in the visible light. To get sensitivity in the visible light, we have here, you see the double bonds. We need a certain amount of conjugated double bonds. Conjugated double bond means that we have a double bond, then the classical single bond, and then again a double bond. So this is a conjugated double bond. Whenever you have conjugated double bonds with a single bond in between, you can finally end up with a translocated electron system which shifts the sensitivity from the UV light to the visible light frequencies. So what's going on, and particularly with the double bonds, you have several or at least two possibilities how they can be arranged. Whenever it looks on different size, this is the trans conformation. But as you see it here it looks on the same side. And this is the cis conformation. When you will count it, you will see that this is position 11, where, or this is position 11, where it gets to the cis conformation. And the main point is now, what's happening when this molecule interacts with light, that you have a conformational change, as I've shown before. The conformation changes from an elf cis retinal into an all trans retinal, because then all of these double bonds, this was the only one in the cis, assume now, this now flips over, and you end up with this flipping over. Here, all are now in the trans conformation. This process is also called a photoisomerization, because isomer, when you check on the formula, the Summon formula, the sum formula, it will not change by this conformational change induced by light. And whenever we have the same formula, we have an isomer. But what's going on, there is a different conformation behind this. So this is a photoisomerization, because light actually switches the cis conformation here into the trans conformation here. And this is a highly, let me say, sensitive process with a very, very dramatic signal to noise ratio. Because in the dark, it will only happen by itself without light in about once per year. Only when light shines on it, it flips over. So it's a highly signal to noise ratio, because in the dark it almost will not happen. And this change in the conformation leads then to a change in the protein conformation. And you can see it here. This is maybe it's MD simulations or crystallographic data. You can see it here. This is the rhodopsin in the dark. Here you can have a look again on how these change actually are happening. You can see here that has been nicely resolved, particularly for scientists who want to have a look on the various states in between. You can see here that occur at a very fast time frame, from picoseconds, nanoseconds to the microsecond range. So in the microsecond range, and we are that's sub-millisecond range, this conformational change is happening. So at least at the receptor level, this is a very fast process. So this is now the flipping over from the cis, and you can see it here, into the all trans conformation. And when we have a look now on the conformational arrangement of a whole protein, okay, of course, we see here the change in the chromophore. You can see it here as well. But what you also can dramatically realize there is a change involved of the overall protein conformation. This change of the overall protein conformation, the protein of rhodopsin is actually called the opsin. This finally leads to a change in the interaction with the G protein, and this then in turn initiates a signaling cascade. So before we switch to the signaling cascade, let's have a look, is there a difference between what we have heard, the rods that are specialized for black and white seeing, and the cones that are specialized for color seeing. So at first, one would assume, let's see, we have here the rods, and here is the spectral characteristics indicated in black. You see a maximum in a range of about 500 nanometers. In the range of about 500 nanometers. And we have three different sorts of cones, one for each color. One for red, of course, which is the furthest part, the longest in wavelengths. So we have in between a green, specialized for green, and the blue, which has the shortest wavelengths in the range of about maximum of about 420 nanometers. And the question is, why do you think that the sensitivity is by these cones is so different? Is there a change in the chromophore which is used here? And that is typically what one assumedly would guess, but the nature has actually used a different approach, because it's too much, let me say, work to generate different chromophores. But what is easily available is to change the primary sequence, meaning the environment of the opsine, of the rhodopsine. And what you can actually see, because the environment of the chromophore is also able to define the absorption maximum. And what you can see here is that there is a change when you compare the primary sequence between these two, between these three rhodopsines, which are actually used for these three different colors, and you can guess where the primary sequence is particularly most different between the blue and the red, because they are so far away in the absorption maximum. However, when you compare between the green and the red, here let me say the primary sequence differs by only 10%, or where between the blue and the red we have a difference by 30% in the primary sequence. So we all have the same chromophore, but we have different primary sequences leading to different environments, and that allows to shift the spectral maximum for the various wavelengths. So just a short molecular medicine approach, or molecular medicine details, color blindness. Interestingly, when all of these three cones are working well, we have a normal color vision, but sometimes it occurs that there are red-green or yellow-blue color blindness. Interestingly, these are much higher pronounced. 80% of men suffer from red-green color blindness, only 1% of women, and particularly when all three are not working, then you have a monochromat. Then we only see black and white. So the cause is that the people are actually missing some of these cones, and sometimes people usually realize it quite lately, because usually when you are red-green blind, usually you will not experience it so easily, but in the next you can see a test where you can see where problems actually arise for people who have red-green color blindness. You can see here the first should be seen by everybody, that's 25. But here it gets different for a person that suffer from red-green color blindness. 29 or 45 are looking only as spots. This is not visible here, but this should be 6 and 8. Also for those who suffer from red-green color blindness, they will see only spots. 5 should be distinguishable, but as long as you involve a green and a red, you also should only see spots. Interestingly, in one of the last year, we had 1 or 2 persons, I would say out of about 50, who realized, although they didn't know it before, they seems to have a short problem with red-green color blindness. But you see, it's not really a problem, because usually maybe you have a different seeing of colors, but usually you will only recognize it quite lately, not at a very early age. So the question now is, when rhodopsine gets activated by this conformational change, we have already heard at the beginning This leads to a closure of sodium channels, and because of the missing sodium influx, these cells in turn hyperpolarize. The question is, what is in between the receptor and which finally leads to a closure of the sodium channels? Most of the cascade, particularly the beginning, and you see here two different images. That one is correct with its localization. Maybe that one is a little bit better in cartoon, but it's not correct because here it seems that all is happening in the same plasma membrane. But you can see actually the signal starts in the plasma membrane of these intercalated discs. Here we have a receptor. It gets activated. When it's activated, we have a change in the conformation, and the conformational change is transduced in turn to a G protein. We will later on see how the activation mechanism of a G protein coupling actually looks like. Let's now move on. What's finally closes the channel, and that is shown here. The channel actually is into the outer plasma membrane, is a compound which we already have heard. It's a second messenger. It's cyclic GMP, and it seems that as long as cyclic GMP is sitting on the sodium channel, as long as it's sitting there, the sodium channel is open and you have a steady state sodium influx. But now, due to the activation of a G protein, an enzyme gets also activated, which now hydrolyzes cyclic GMP to GMP. And this leads to a decrease in the cyclic GMP concentration, which in turn leads to a dissociation of cyclic GMP from the channel, and then leads to channel closure. So on the next slide, you can see what happens after the conformational change of the rhodopsin receptor. Here, the rhodopsin receptor actually gets activated by a ligand, and the ligand in our case is light, makes a conformational change which increases the affinity to the G protein. And the G protein, maybe I forgot to mention it before, the G protein here is one of the first G proteins identified. It's called transducin. And that the name tells what it is. It is from the Latin word transducere, meaning it somehow helps the signal to transmit from the conformational change of a receptor to the activation of an enzyme we will hear in a second. So as any G protein, also transducin, is composed of 3 subunits, and they are termed after the Greek alphabet: alpha, beta, gamma. And you can easily guess what's the most important. Of course, the most important subunit is the alpha subunit. The alpha subunit actually is the subunit which interacts with the activated transducin. That's the receptor for light. And when due to the conformational change, the alpha subunit activates with a receptor, with a rhodopsin, we have a change in the conformation transformed onto the alpha subunit of a G protein. And you see in the resting state, the alpha subunit has a GDP bound. So that's the reason where the name G protein or coupling proteins, the name G comes from, because actually guanine nucleotides are binding in a certain way to the alpha subunit, which is responsible for switching from an inactive to an active state. So in an inactive state, we have guanosine diphosphate bound, which exchanges because when the alpha subunit interacts with the activated rhodopsin receptor, then we get a change in the conformation also of the alpha subunit of the transducin, so that the affinity for binding of guanine nucleotides is now decreased for GDP and is increased for GTP. So we always have GDP as well as GTP in the cytosol available. But this depends on what has the higher affinity? And now when the transducin, particularly when the alpha subunit comes into an activated state, then it binds with a higher affinity, as you can see here, GTP, guanosine tris phosphate. And this marks that the G protein, particularly the alpha subunit, is in activated state. And with this binding of GTP, there is another process concomitantly occurring, that is the dissociation of beta and gamma. And in 95% of all cases now, the activated alpha subunit with G protein bound further activates effector systems like, we have already heard, second messenger, adenylate cyclase, generating cyclic AMP. In our case, it's also not phospholipase, but is, as it's written here at the end, phosphodiesterase, and particularly a cyclic GMP dependent phosphodiesterase, as we will see in a second. So let me just finish how this G protein, the activated form, is actually inactivated. After some time, these alpha subunits, when they interact with the effector systems, they have an inherent GTPase activity going on. And with this GTPase activity, after some time, GTP is hydrolyzed. So a phosphate is clipped off. And what's remaining finally is, because a phosphate is clipped off, is again a GTP. So here it's not an exchange, it's an hydrolysis of GTP into GDP. And with this now, when GDP is sitting here at the alpha, it associates again with beta gamma. And here we have now, in a very cyclic way, the resting state of a G protein, which is available now for a second cycle, for another cycle, getting activated due to an activated rhodopsin receptor. So here is just simply in a sort of a movie how this actually happens. You can go through it, but it's the same as what I've just told you right now. The main point is the interaction of the activated alpha subunit of the transducin occurs with the phosphodiesterase. And phosphodiesterase now, it hydrolyzes cyclic GMP into GMP. Why is it called phosphodiesterase? In the GMP molecule, particularly in the cyclic GMP, we have a diester. And the diester, that is hydrolyzed, ending up in a monoester. So that is phosphodiesterase, because the ester actually comes from the phosphoric acid, so it's a phosphodiester, and that is how to understand the name phosphodiesterase. We have heard that an ACE usually is usually an enzyme which somehow hydrolyzes compounds, which splits compounds. And diester is splitted here, so that's actually a second point. And the third point is it's a diester of a phosphoric acid, so this matches particular requirements for cyclic GMP hydrolysis into GMP. Let's have a closer look on the interaction of rhodopsin with the G protein, with the transducin that has been already identified, particularly some strands. So this is the alpha subunit, the beta and the gamma subunit of the transducin. This is, in particular, it's a cytosolic protein. It's not a transmembrane protein, in contrast to the rhodopsin receptor, which has these 7 transmembrane helices. But you can see here that the G protein is tacked to the membrane by myrostyl or farnesyl groups, which can be used for derivatization of amino acids. These are groups that have a high hydrophobic moiety, so they can easily integrate to one layer of the plasma membrane, and this allows a sort of fixation at the intracellular side of a plasma membrane for the G protein, transducin. And you can see there have been already strands identified in the rhodopsin here and at the C terminus that interact particularly with the alpha subunit in this process of activation, which finally leads to the exchange from GDP to GTP and the dissociation of alpha from beta gamma. So with this phototransduction process, there's also really a very impressive amplification going on. So we have heard of this high signal to noise ratio, particularly as the noise is very low, but where comes the signal from? We have one rhodopsin molecule absorbs one photon, so it's very high sensitivity. In the interaction, as long as the rhodopsin is activated, it can activate 500 to 1000 transducin molecules. We have heard one transducin activates one phosphodiesterase, but the phosphodiesterase, when it gets activated, as long as the transducin in the activated form, the GTP carrying alpha subunit, stays on the phosphodiesterase, we have a 100 to 1000-fold stimulation of phosphodiesterase activity. So on the whole, there's an amplification by 10 to the 6. So this really is responsible for the signal to noise ratio with this phototransduction process. So let's have a second look here what happens when the cyclic GMP concentration decreases, because due to the activation of the phosphodiesterase, that is what we've heard at the beginning, the sodium channels, they close. So the sodium influx actually is missing. We have a missing of positively charged entering the cell, and this leads in turn to an hyperpolarization of the photoreceptor cells. And this is somehow linked. You can see here, we will come to that in a second. Here's the process as we had at the very beginning. In the dark, we have a depolarized state at around -30, -40 millivolts. -40 millivolts, as you can see here, when we measure the membrane potential. When light shines on those cells, you see this hyperpolarization process. And what you can see here, this is actually the current measured through the sodium channels. This reflects what's going on with the membrane potential. It hyperpolarizes because the current actually stops here. The channels get closed, so we miss the sodium influx. But at the same time, because these channels, we see it in a second, we will not only allow an influx of sodium, it's 90% sodium, but 10% is calcium. And this is coupled to a transporter, a sodium, potassium, and this is here the sodium potassium transporter, but now a special transporter comes into place. We will come to that in a second. Let me have a closer look on the structure of this channel. Meanwhile, maybe it has been already crystallized, but I want to mention here is you can see here there are different forms available, but they have one similarity in the C terminus. This is the CNBD, the cyclic nucleotide binding domain. So here actually in the C terminus is that domain where cyclic GMP is bound. And the main point is when cyclic GMP is hydrolyzed in the cytosol, you see a decrease in the cytosolic concentration. So the equilibrium is shifted by dissociation of cyclic GMP from the channel, and then in turn the channel gets closed. You see here it's classical 6 transmembrane helices. We do not have a voltage sensor here because that's a classical ligand-gated channel. The ligand is cyclic GMP, but of course we do have a pore, we do have a selectivity filter. So there have been some electron microscopy images. Maybe meanwhile we have a better resolution, but what you can see here, this is maybe a side view, And this is a view from above, that in the middle here you can see here a centralized pore, and it seems that this pore is actually where sodium is flowing into sodium, and also a certain amount of calcium is flowing into the channel. But this resolution of the electron microscopy here is in the tenth of micrometer range. So what are mechanisms to somehow shut these processes off? And of course, one we have already heard, that is the hydrolysis of GTP on the alpha-1 subunit. That is the inherent GTPase activity on the alpha-1 subunit of transducin. After some time it gets active, GTP splits off due to GTPase 1 phosphate, and it ends up with GDP on the alpha-1 subunit, which we associate with beta-gamma. The second actually takes place at the level of rhodopsin. So even the transduction or the transmission from rhodopsin onto the G protein can be stopped, and we will have a process going on that the cells in the dark can get back again to the depolarized state. Let's first look at the second step of shutting off the system. At the level of rhodopsin, there actually a phosphorylation can take place, and the receptor, or let me say the phosphorylation occurs via the rhodopsin kinase. So the rhodopsin kinase via ATP puts some phosphate groups, you see it seems on the C-terminus. That is the site which we have shown before that interacts as one of the important sites with the G protein, with transducin, with the alpha-1 subunit. But that's not enough that here a phosphate group or two phosphate groups are put on. The second actually comes from another molecule which associates and somehow shields these groups here from an interaction with transducin, and that protein is called beta-arrestin. To put something under arrest, it's very clear what it means. You shut somebody off, and that is what is occurring here. Rhodopsin actually is shut off from its interaction with the alpha-1 subunit of a transducin. So the next process has been identified quite lately because it involves a very interesting exchanger which is working all the time in these photoreceptor cells. Because you can imagine when in the dark state, these sodium channels that not only transport sodium but also about 10% of the influx comes from calcium. So this would finally end up, when these channels are open in the dark state, in a very high calcium concentration. And as we have heard before in the topic with the negative feedback mechanism with calcium channels, here it's also important to somehow limit the increase in the cytosolic calcium concentration. So here all the time an exchanger is working, and you see the exchanger is exchanging sodium. This provides the driving force because the energy, because we have no sodium almost, or let me say a much smaller concentration of sodium inside of a cell. And this allows in exchange that calcium and potassium can be pumped out of a cell. So this somehow limits in the dark the cytosolic calcium concentration to quite a high level, but otherwise without these transporters it would end up much higher, and maybe it would lead even to damages of the photoreceptor cells. So we have a concentration of about 500 nanomolar due to this exchanger. Now when these channels close, because cyclic GMP is hydrolyzed and dissociates, this exchanger is going on, and you see it's effectively going on, because what's happening, the calcium concentration now drops down to a very low level. It even drops below to classically resting cell calcium concentrations, which are in the range of about 100 nanomolar. So this drop in the calcium concentration, now a certain protein comes into play which actually gets active when the calcium concentration gets so low. You may remember, as we've heard in the first topic, Calmodulin is a calcium binding protein which gets activated when the calcium concentration increases. This protein, and it's called recoverin, gets actually activated when the calcium concentration decreases. And the name of this protein, recoverin, tells us what it does. It helps to recover from this hyperpolarized state, getting back to the resting state with a sort of depolarization. So what's going on here? We have a cyclic GMP, which is hydrolyzed. It leads to the close of a sodium calcium channels. This in turn, these calcium concentration gets smaller and smaller due to the exchanger, and here a recoverin without calcium, because it loses the calcium, gets activated. And the main point is, and when you think it's that the mechanism must work quite simply, the recoverin, the aporecoverin, meaning in the absence without calcium, interacts with the guanylyl cyclase or guanylate cyclase, and guanylate cyclase is the enzyme which produces from GTP now again cyclic GMP. So as you can see here, very interesting, also a cyclic mechanism in terms when the light shuts down or closes the sodium calcium channels. When the light is now switched off again, we need to reopen the sodium calcium channels, and they can be reopened by an increase in the cyclic GMP concentration. That is actually what is happening here. Recoverin activates guanylyl cyclase that now synthesizes from GTP cyclic GMP increases in the cytosol, it binds again to the sodium calcium channels, they get opened again, and we have this steady state influx of sodium and calcium which leads in the dark to the depolarized state. So let me now here present an overview on the mechanism, and here you can correctly see where these processes take place. We start here with the rhodopsin that gets activated by light. When it's activated rhodopsin, the signal is transmitted to the transducin, which dissociates with an activated alpha subunit of a transducin with a GTP bound. This activates phosphodiesterase. The phosphodiesterase now in turn hydrolyzes cyclic GMP to GMP. And you can see here this channel, 90% sodium, 10% calcium, is only opened in the presence of cyclic GMP. When cyclic GMP drops, the channel closes in turn. And we have here a transporter going on which exchanges sodium for potassium and calcium. So we have here, when these channels now are closed, a further decrease to very small intracellular calcium levels. And this in turn activates, you see the decrease in the calcium concentration. Protein, it's maybe not nominated here, but that is recoverin, and this recoverin activates the guanylate cyclase, which in turn from GTP synthesizes cyclic GMP. Cyclic GMP increase in the cytosol, binds to the channel, and the channel gets reopened again, leading to the steady state influx of positive charges, and we end up with a depolarized state. You can nicely see here that all what is connected to the rhodopsin happens in the intercalated discs. What is connected to the cyclic GMP channel is happening actually in the outer plasma membrane. Let's now have a final look what's going on finally in the transduction of these processes, signal transduction to the visual cortex. I will have a short look at this because it's a very complicated process. You can see the visual cortex here is at the very end of our brain, so behind. And you can see here, starting with the optic nerve, which is gathered here and leaves all connected. There is one relay where the signals are relayed here in a ganglion. We will see it in a second, and from here it ends up in the visual cortex, where actually an image is composed so that we can see based on actually the light that falls through the eye. This nucleus, which has been shown before, is called the nucleus geniculatus, or lateral geniculate nucleus. This is where we have a switching of the signals, and let me point out one maybe important process where signals are projected, visually projected, because at least what I've heard, but I misunderstood it, that all what you see on the left eye is finally projected to the right. But when you look here, that's not correct. What's correct is all what you see on the left side finally ends up in the right part of the visual contact. Let me just focus it here. You can see that is the left side here. The left side, you see it on that part of the retina and on this part of the retina. And when we now follow these two, you can see here, these two, they get together on the right side. So all what we see on the left finally ends up on the right side of the visual cortex. And vice versa, it's true, all what we see on the right side ends up on the left side. So what do you know what's going on here? And there have been experiments done with a microelectrode inserted in the retina to somehow detect these electrical signals. Of course, the animals have been somehow get a narcotic so that they do not have any hurt. And you can see here there have been come up the hypothesis of the receptive fields. So there are a certain area in the retina which is called a receptive field, and the receptive field finally ends up with a certain stimulus. So let me just introduce the concept of receptive field. So we have in the background, we have a certain action potential frequency. And when now we illuminate a certain area here in the center, so this is the center illumination, you can see here that it's an increase in the frequency. However, when we are outside this center, but we are still in this receptive field, when the illumination is outside of the center, then we have a decrease in the frequency of action potentials. And when we are outside of a receptive field, then nothing is changed. So it seems that the signal that is finally generated through photoreceptors in a sort of receptive field, and it finally ends up in the ganglion cells, that somehow shows sensitivity whether we have a center illumination or an illumination outside of the center but still in the receptive field. So here in addition we have two types of cells. The one are called off-type bipolar cells, the other are called on-type bipolar cells. There is one main difference here. Whenever we have a central illumination, the off-type bipolar cells, they are in a depolarized state, which finally gets hyperpolarized, while the on-type bipolar cells, they are in a hyperpolarized state, but they in turn get, you can see it here, they in turn get depolarized. So this is a very clear distinction between off-type bipolar and on-type bipolar cells. You can see here, these bipolar cells are linked to a number of photoreceptor cells which are placed in this receptive field. So with an on cell, light with a central illumination produces depolarization. With an off-type cell, light to the central illumination produces hyperpolarization. And the other way it's round, when we have not the central, but we have light surrounding regions, this produces hyperpolarization, and with an off-type cell, this produces depolarization. So let me just see how these signals are further transmitted from the bipolar cells. We have heard later on the bipolar cells are connected to the ganglion cells. And you can see here the signal transmission looks the same. What happens in the bipolar cell also happens in the ganglion cell. But there's the main difference. You can see here in the cones or in rods, we always have a hyperpolarizing signal. But with the off-center bipolar cells, and that is only true here for the central illumination, we have a hyperpolarization that is also transmits to the ganglion cells, also with a hyperpolarizing signal. However, with the on-center bipolar cells, we have a depolarization, and that is transmitted to the on-center ganglion cell, also in a way of a depolarization. So let me show you now an understanding of this different regulation of the off and the on type bipolar cells. Maybe it's a little bit complicated, but let's first have a look on the off-center bipolar cells. You can see here in the dark, these nerve cells are depolarized. So depolarized cell means we have actually a release of neurotransmitter, and you can see here the neurotransmitter involved is in all of these cases independent of on or off type receptor cells is glutamate. So glutamate is binding to a receptor. This receptor is a ionotropic receptor. At the same time, it allows a steady state sodium influx. And you can see here, this sodium influx would actually lead in the dark to a depolarized cell. Now, under light, these cells now hyperpolarize. Because they hyperpolarize, there is a stop of a glutamate release in a hyperpolarized cell, because it gets close to the resting state. So there is no action potential occurring, so there is no release of glutamate. No release of glutamate means the depolarization now is shut down. There is no influx of sodium. This leads in turn, because due to the missing sodium influx, to a hyperpolarized signal. Off-center bipolar cell as well as the off-center ganglion cells. When light shines on those off-center cells in the central illumination, these cells, they hyperpolarize. So how is it possible that with the same neurotransmitter, exactly the different case occurs with these on-center bipolar cells? Here in the dark, glutamate, as we've heard before, is released. Here we have a different glutamate receptor. And you see, this glutamate receptor is coupled via a G protein to another channel. And this channel is a TRPM1L channel. It comes from the transient receptor potential ion channel family. And this channel, as long as glutamate is sitting here because it's released via a G protein, this channel is closed. So we do not have here an influx of sodium, particularly sodium. Sodium goes through these glutamate receptors here, but it also would go through that channel. But that channel is closed because due to a negative regulation via this G protein. So this channel is closed. So these channels actually, in the dark, they are in a hyperpolarized state. But when now light shines on it, then we have a stop of a glutamate release, because there is no released or a decreased glutamate concentration. The negative regulation via G protein or this channel stops. And in turn, because the negative regulation of a G protein is missing now, this TRP channel open. So in the light now, because of the missing glutamate, these TRP channels open, they allow a sodium influx, and in turn we end up with an on-center bipolarized cell in a depolarized state. And the same happens here. When it's depolarized, here we also have an on-center ganglion cell which also depolarizes. So particularly this channel is so important for making the difference between an off-center and an on-center bipolar cell. So are there further proofs? And this indeed has been proven by a knockout animal, where people have shown that here, when this TRPM1 channel is present, and you can see here it's also localized here in the retina by an antibody staining, and when light shines on it, these channels get open because the negative G protein regulation due to glutamate, which would actually put this channel in a closed state, is missing now. So we can see here an influx of sodium that leads to a depolarization in these on-center cells. However, in a knockout animal where these channels are completely missing, and you see it here in the staining, when light shines on the same type of cells, but that are missing now the TRPM1 channel, you do not see any opening of currents, you do not see any depolarizing influx of sodium. So that was a further proof that this channel is so important for the difference between off and on type receptor cells. So the rest gets extremely complicated. From a receptive field, let me just only say this is the nucleus geniculatis, as we referred before. Here is a further signal relaying. So interestingly, black and white is somehow separated in the axons from the color seeing, and at the end, at the visual cortex, where the final image is put together, I'm not sure whether this is already solved, how this works, but it's a highly complicated process and would go beyond the scope of that lecture. Just one look on how many photoreceptors actually we have. You see the humans have a really high amount of photoreceptor cells, but don't underestimate the housefly, also 80,000. Interestingly, spiders, some of the spiders have 10,000, and particularly in algae there is one photoreceptor present. So there are very different amounts, but you can see here the largest amount of photoreceptor cells actually are present in humans. So this is the end of the signal transduction process, and I'll hear you again in the next topic 3.